Today we'll be starting with complete and incomplete dominance. In the last class, we had this basic idea of the Mendelian laws, and from the Mendelian laws, one of the deduction was the concept of dominance and recessive. Now, if you look into this picture, let's move into the next slide. So as you can see, there are two alleles, A1 and A2. When A1 is in a homozygous condition, we get a red flower. When A2 is in homozygous condition, we get a white flower. The question is, who is dominant over whom? Whether A1 is dominant over A2 or A2 is dominant over A1. So to understand the dominance, we have to look at the phenotype of the heterozygote. So if there is an A1 allele and A2 allele, depending on what the color of the heterozygote, remember, the A1 gives red color and the A2 allele is giving us white color. In homozygous condition, A1 are red flowers. In homozygous condition, A2 allele gives white flowers. To understand whether A1 is dominant over A2 or A2 is dominant over A1, we have to look at the heterozygous flowers. If the heterozygous is a red flower, then we can easily assume that A1 is dominant to A2. But again, if the heterozygous is a white flower, then we can assume that A2 is dominant to A1. So from this we clearly understood, in fact, in yesterday's cross also we again came up to this point that the phenotype of the heterozygous actually tells us which is the dominant allele. Now, so while doing Mendel was lucky in the sense that there was this phenomenon of complete dominance in case of pea plants. One allele is completely dominant over the other allele and is completely masking the expression of the other allele. But in certain scenarios, there are phenomena which are referred to as incomplete dominance. In incomplete dominance, as you can see, the phenotype of the heterozygous falls between the phenotypes of the two homozygotes. So if this is the example, and we assume that A1 is dominant to A2, so the red flower is dominant to the white flower. So this is the A1 homozygote, and this is assuming this to be the A2 homozygote at this end, and we are having the A1 homozygote at this end. You can see the range, phenotypic range of the intermediate flowers. That means the dominant allele is not successful in masking the expression of the recessive allele. And when such phenomena occurs, we observe the phenomena of incomplete dominance, where the dominant allele is not completely dominant over the recessive allele. A beautiful example of such a case can be seen in nature. So this is eggplant. We have a purple fruit and we have a white fruit. And for the purple fruit to be dominant over the white fruit, the F1 heterozygote should be purple in color. But after the cross and the fertilization is done, as you can see, the fruits are violet. So the fruits neither are similar to the purple fruit or to the white fruit, but is showing an intermediate expression, which clearly indicates that the capital P for the purple allele is not completely dominant over small p, the allele for the white color. And evidently, when we go for the final F2 generation, what do we see? We see purple, we see two violet colors and one white. If you remember the Mendelian ratio was 1 dominant homozygote, 2 the heterozygotes, and 1 the recessive homozygote. So In case of Mendel, since this too phenotypically gives us the characteristics of tall plants, so 3:1. In case of incomplete dominance, we are seeing that the heterozygotes are distinctly different from either of the homozygotes. So phenotypically, it is 1:2:1. Genotypically also, it is 1:2:1. So from outside, we can distinguish the dominant homozygotes from the heterozygotes, and hence we are getting a phenotypic ratio of 1:2:1. Similarly, genotypically, obviously they are different. It is 1 dominant homozygote, 2 heterozygotes, and 1 recessive homozygote. So in case of incomplete dominance, we observe the phenomena where the phenotypic ratio becomes equal to the genotypic ratio. Excuse me. Now, let us come to the definition of this dominance and incomplete dominance. As I said, and again, we saw that phenotype of the heterozygote is same as the phenotype of one of the homozygotes. So the homozygotes with which the phenotype of the heterozygote is similar, that becomes the dominant allele. In case of incomplete dominance, what do you observe? The phenotype of the heterozygote is intermediate, falls between the phenotypes of the two homozygotes. That we observed, and we saw the case of eggplant. Finally, there's another concept which is codominance. In the concept of codominance, both the alleles are expressing equally. There is no blending of the color. In case of eggplant, what we saw? There was a purple flower, there was this white flower, and we saw blended intermediate violet flower. But in case of codominance, the colors do not blend. In case of codominance, the heterozygote includes the phenotypes of both homozygotes. We'll come to that example. But before that, there is another concept I'd like to give. It is that when we talk about a phenomenon of complete dominance, what do we mean? For example, this is exemplified. This is capital P, capital P. So this, both the alleles is producing a functional P protein. Since there are two alleles, 100% of the product is being produced, and we are getting purple color. I'm talking about simple dominant recessive ratio. I'm not talking about incomplete dominance or codominance. Now let us look at the heterozygote. There's only one single capital P. So the single capital P is producing 50% of the protein product, and that 50% of the protein product is sufficient to give a purple color to the flower. In absence of the capital P, there is no P protein product, and thus we are getting, whatever we are getting is a recessive phenotype. Now, in this particular two cases, as you can see, 2 doses of capital P is giving me a purple color. 1 dose of capital P is also giving me a purple color. So those genes who in 1 dose gives an expression equivalent to the expression which they gave in 2 doses are known as haplosufficient genes. That means the purple gene, 1 dose is enough and is equivalent to, and the output of the 1 dose is equivalent to the output of the 2 doses. So most dominant genes which are capable of perfectly masking the recessive phenotype are haplosufficient genes. So their 1 dose is enough to give an output which is equivalent to the output of had there been 2 doses of that particular gene. So now, since we have finished with haplosufficient, we will move on to the various types of alleles that you observe in nature. Just a second. Now, as I said, we'll move on to some concepts: multiple alleles, isoallele, pseudoallele, lethal allele, and allelic series. The first one that I'll start is a multiple allele concept. And while doing this, we'll understand also the phenomena of codominance. We studied complete dominance. We studied the case of incomplete dominance. By definition, we studied the phenomenon of codominance, but now we'll understand what exactly codominance means. And the beautiful example of both multiple alleles and the phenomenon of codominance can be seen in our blood group. We all are aware that there are 4 basic categories of blood group. Blood group A, B, AB, O. Keep in mind that blood group A is coded by 2 alleles, IA, IA. Blood group B is coded by again 2 alleles, IB, IB. This is how it's symbolized and written. When we have AB, so this is codominant. So there is one IA allele and one IB allele. Both are retaining their features. And finally, there is this recessive allele that is often written as small I. Okay. So since if you look into the dominance hierarchy of the blood group, IA is codominant to IB. So they are equal, but both of them dominant to small I. So if both of them are dominant to small I, then if we have a combination of IA and small I, that will also give A blood group. If we have a combination of IB, small I, that will also give a B blood group. So if you can see the genotype of A blood group, it is IA, IA or IA, small I. B blood group, IB, IB or IB, small I. AB blood group, IA, IB. And finally, O blood group, small I, small I. So that is how the alleles are arranged and what the expression they give. Now to keep it in mind that how is this that these alleles are controlling the blood group? Keep it in mind that the IA and IB alleles actually they encode an enzyme known as glycosyl transferase. These enzymes recognizes the sugars. This enzyme actually, let me not put it this, let me put it this way, that they add sugars to the terminal chain of a protein found on the surface of RBC. So in the surface of RBC, there is a molecule and they add terminal sugars to this molecule. Now, how this happens, let us have a brief view on the molecular mechanism of this occurring. Now, let us look into this. The wild type FLUT1 allele is present in all humans. And what is the function of this allele? The function of this allele is to add a fucose molecule to a precursor substance. So a precursor substance is present, which is made up of galactose and acetylglucosamine. And FLUT1 allele adds a fucose molecule to this precursor substance to convert it into H substance. So to repeat once more, there is this FLUT1 allele present in all humans, which helps in the conversion of a H substance precursor to H substance by adding fucose molecule to it. So you can see that once fucose has been added to this molecule, we are getting this H substance. Now, as I said, both IA or IB allele can code for an enzyme known as glycosyl transferase. And these enzymes now add a terminal sugar to this H substance. If it is the IA allele and the enzyme that the IA allele codes is glycosyl transferase, and this glycosyl transferase then adds an acetylgalactosamine to the H substance. remember, just if I go back, the O blood group has only 8 substances present in their blood because the O blood group does not have an IA allele or an IB allele. So in their blood, they have this H substance present. For the A blood group, where we have a IA condition, or we have a IA small I condition, we get this scenario. The IA allele produces the glycosyltransferase, which adds an N-acetylgalactosamine to the H substance. Similarly, when the condition is IB, or the condition is IB small I, what happens? A glycosyltransferase is encoded, and this glycosyltransferase will add galactose to the precursor substance. So addition of N-acetylgalactosamine to H substances converts it to a A blood group. That is, A antigen is present on the surface of the RBC, or addition of the precursor H substance, galactose to the precursor H substance, converts it into the B antigen. Remember that both the A blood group and the B blood group has this precursor molecule present in their blood. Hence, if they get blood from O, which contains only the H substance, no reaction occurs because in their body also they have this precursor H substance. And hence O is referred to as the universal donor. Now, when the concept of codominance is coming, in the case of the blood group, let me erase all these things. In case of people having IA, IB, so if there are 100 molecules of these precursor substances, 50 molecules, for 50 molecules, N-acetylgalactosamine will be added because of the IA allele producing the glycosyltransferase. And for 50 molecules, IB will provide the glycosyltransferase, and that will add galactose to the H substance. And thus, the AB blood group will have the A antigen as well as the B antigen on the surface of their red blood cell. Now, this is another picture showing you how in case of O blood group, neither A or B antigen is present on the surface of the RBC, how the antigens are present. In case of A blood group, we have antigen A. In case of B blood group, we have antigen B. And in case of AB blood group, we have both antigen A and B. This is a checkerboard, and this is, you can see, IA results in addition of N-acetylgalactosamine. IB results in addition of galactose as a terminal sugar on the surface of the RBCs. And thus we have this ABO blood group system where we are seeing that the A antigen and the B antigen are present simultaneously on the surface of RBC if IA-IB allele combination is present. That means IA and IB are equally expressive. So this was a beautiful example of codominance. Now I'll show you some of a dominant hierarchy series. Let us look into this case. Now there are 3 alleles associated with this duck pattern. One is the M allele, the other is the MR allele, and the third is the MD allele. So there are 3 alleles. MR produces a pattern called restricted. M produces a pattern called mallard. MD produces a pattern called dusky. Now remember, even if though there are 3 alleles, for a given chromosome, any two allelic combination will be present to give a phenotype. So it will be any two allelic combination from these three alleles. Now, as I said, these are three alleles and there is a hierarchy, dominance hierarchy. That means MR is dominant to M, is dominant to MD. So this is the hierarchy. So if we have a combination of MR, that is, we'll have restricted pattern. Again, MR, M. Since MR is dominant to M, we'll also have a restricted pattern. Finally, MR dominant to M. So if we have a combination of MRM, we'll also have a restricted pattern. Since any allele that comes in the combination with MR, either M or MD, will have a restricted pattern. Again, if we have a combination of M, MD, we'll get a mallard pattern. We'll get a dusky pattern only when we will have a combination of MD, MD. So remember that whenever there is a dominance hierarchy, that means MR is dominant to M, is dominant to MD. The phenotype of the dominant allele will be expressed because we are doing an example of dominance hierarchy. No incomplete or codominance is seen here. And since this is a definite order in which the dominance is following, so the phenotype of the dominant allele will be seen. Now let's look into the write-up just a second. See what happens with the cross. So when we do a cross like this, as I was talking about, that the three R's, MR restricted, MD dusky M produces the mallard pattern and MD produces the dusky pattern. So when the cross occurs like this, what are the combinations we can have? MRM, MRMD, MDM, MDMD. Now see, phenotypically now see. MRM and MRMD will be restricted because MR is dominant to either M or MD. M being dominant to MD will have a mallard pattern and MD will be dusky. So this was what I was telling you, that when we are having a hierarchy of MR dominant to M, dominant to MD, all the combinations with MR will show restricted pattern. All the combinations with M will show mallard pattern. And the combination with MD will show dusky pattern. So there is a formula that if N is the number of alleles present at a locus, then what will be the possible number of genotypes that can be obtained, which is given by the formula N into N plus 1 total bracket divided by 2. Given that you know what are the number of alleles present at a locus. For example, in this case, we have 3 alleles. So you just put 3 here. 3 into 3 plus 4 for 12 by 2, 6. See, we are having 6 genotypes. So this is a generalized formula for the fact that if now a formula for the number of genotypes that can be obtained for a given number of alleles at a particular locus. Remember, while working crosses with multiple alleles is very similar to one when you do it with 2 alleles. The Mendel's principle of segregation still holds because the alleles are being segregated independent of each other. And finally, now we move on to another part, that is isoallele. When I'm talking about isoallele, we move on to the eye color of Drosophila. In this example, what we see that Drosophila in multiple alleles in Drosophila occurs for one gene pair affecting the eye color. Now, the recessive one is WW, white. And the dominant eye color is W plus W, that is red. These are the two extremes. Now, but when scientists and researchers did a detailed study and extracted detailed study on the measurements of eye pigments, this kind of table was obtained. And it was seen that when we are having a relative amount of pigment around this range, the genotypes appear as non-wild. See, all of them are not WW. You can see the change in the symbols, but all of them are phenotypically showing white eye color. Similarly, interestingly, if we look into these three cases that I am showing you in blue boxes, these three cases, you can see the number of relative amount of total pigment is different. The symbol is also different, but all of them phenotypically exhibited red color. Red color is given by WW. Okay, so all of them are phenotypically showing red color. So that is where the concept of isoallele develops, where isoallele says that these allele acts within the same phenotypic range of each other. WW is different from W plus S, W plus S. In the very subtle difference in the amount of relative pigment, sorry, in the amount of total pigment present in the eye, but yet phenotypically they are red in color. So this is important. That is, the alleles acting within the same phenotype range of each other are known as isoalleles. So the three blue boxes that have been highlighted here are isoalleles within the normal phenotypic range. Similarly, there are certain phenotypes which also show white colors, mutant phenotypes which also show the recessive eye color trait. So then we have the mutant isoalleles. Just remember that in case of a multiple allele system, if the alleles in different combination shows the same phenotypic manifestation, then we have to assume that these alleles falls under the isoalleles. And finally, for today, we'll have our last concept, that is the concept of pseudoallele. When we are talking about pseudoallele, the pseudoallele was studied by MM and KC Green. And when we consider the case of pseudoallele, first of all, you have to remember that allele is a position where a gene is sitting on a chromosome. In case of pseudoallele, these alleles are present in complex locus. That is, their position can be divided into a number of subloci. You can see this is the position where the lozenge gene is sitting, but the lozenge locus consists of 4 groups. So this is the lozenge locus, LZ. This lozenge locus consists of 4 groups. You can see presence of 4 groups in the lozenge locus, and each containing one or more different sets of alleles. So just remember, this whole locus is the lozenge locus, and the lozenge locus consists of 4 groups, each containing one or more different lozenge alleles. Now, in such a setup, what was observed was, you can see that you can observe that when the mutants are present in cis position, plus is the wild type allele, and the names that are written lozenge BS, lozenge K are the mutant alleles. When the mutants are present in the cis position, that means they're present on the same chromosome, we are getting wild type phenotype. But whenever the mutants are present in transposition, that is, one mutation is one chromosome and the other mutation in another chromosome, then we are getting a mutant phenotype. So initially, when this was studied, so I was talking about so just compare the corresponding the lozenge BS and lozenge K in cis position, lozenge BS and lozenge K transposition. In transposition, we are getting mutant phenotype. In cis position, wild type phenotype. It's same for all. So it is observed that mutations within the same gene locus on both homologous chromosomes prevent either chromosome from producing the normal product. And this phenomena, recombining closely linked alleles, were named as pseudoalleles. That was widely used during that time. Because why were they called pseudoalleles? Because you can understand the lozenge is, as I said, is a single locus. Within this locus, we are getting 4 subloci. And mutations are occurring within the subloci. If mutations fall in the same chromosome, no problem, wild type. If mutations fall in different chromosomes, then we are seeing that somehow there is a prevention of forming the wild type phenotype. So this was the concept of pseudoallele. As I said, it consists of subloci, and this was a conclusion. Even when recombination is possible between the alleles, so if the mutations within the same gene locus on both homologous chromosomes, so the mutations are present on both the homologous chromosomes, and even when recombination is possible between the alleles, however, this presence of mutation on both the alleles is preventing either chromosome from producing the normal product. And with this, our last slide of today's class is the allelic series. Now, look, we have studied one allelic studies. That was the Mallard duck, where there were 3 alleles. See here, we have a C plus allele, we have a CH allele, we have a CH allele, and we have a C allele. So when we are having alleles like this, so in this case, the dominance hierarchy is C plus allele, dominant to CCH, dominant to CH, dominant to C. So all those combinations with C plus, all the combinations with C plus will show wild type. Since CCH is dominant to the other 2, anyway, what has been shown here is CH is dominant to small c. CCH is also dominant to CH. In either case, we will be getting light chinchilla. Look at the color. Again, it's light chinchilla with black tips. When you're having a CH presence of this, there is somewhat presence of black tips. So this CH allele is in fact not being totally masked, or there is other phenomena occurring. And finally, when we are having this combination CHC, we are getting a Himalayan type. So as you can assume that when we have allelic series, it is important to understand which is the dominant allele, and depending on how the dominance hierarchy is, the different phenotypes are accordingly exhibited. As I was saying, that the wild type allele is completely dominant of all other alleles, but the chinchilla allele, so the CCH allele is partially dominant over the Himalayan and the albino allele. And hence we get this formation of black tips. So there is not complete dominance when we are considering the chinchilla alleles. So with this, we come to the end of the different concepts on alleles. There is one more concept on allele I need to give before ending today, that is the lethal alleles. For example, let us look into these 2 moms. One is AY and the other is AL. AY A plus and the other is AY A plus. So this is a heterozygous condition. Body color yellow. Now, what do we consider as lethal mutations? Mutations that interfere with necessary vital functions are called lethal mutations. And if there is a lethal mutation present in a homozygous condition, then that individual dies off. In case of dominant lethals that occur in early in life, and since dominant lethals are, as you can understand, dominant, So most of them die very early in life. Whereas in case of recessive allethals, they remain hidden for some time. But ultimately, if a homozygous condition is achieved, then this recessive lethal is expressed and people, I would say, the organism dies. So let us look at the cross. So we have a simple Mendelian monohybrid cross. So you can see AY was the lethal allele, and we have of the four F1 generation, what we are for F2 generation, what we are achieving is the 1/4 of the generation is having AY lethal allele in a homozygous condition, and they are dying off. So this was about lethal alleles. And from next day, we'll do the cis-trans test for allelism. We'll do it next day. We'll continue our class next day from here.